Hey guys, this is Kendall Terry, and this is cell division part 2. Before we looked at mitosis, and now we're going to look at kind of what takes place to make sure mitosis happens, and also the proteins involved that promote different phases along the way. So we see here kind of like an overview of the whole process getting started. We've got some kind of growth factor that will come in here and bind to this receptor. Now this is a tyrosine receptor kinase, a TRK. Alright? And it's been autophosphorylated. You can see that here with the phosphates attached. Those are docking stations. RAS protein is going to come in and bind to that protein, or that phosphate there. That will activate RAS. RAS will cause a MAP kinase cascade to take place. When that MAP kinase cascade takes place, it will produce a protein that will come into the nucleus and start to encourage transcription factors that will promote genes that will be made into proteins. And the protein that you see here at the very beginning is the G1 cyclones. As the G1 cyclones are made, that begins the whole process of cell division. So this cell right here would be in the G1 phase, and as all of this takes place to make these G1 cyclones, that's going to kick it into going into the next phase. So let's go ahead and look at that. So we see here that we've got the G1S checkpoint. Right there at the end of the G1 phase is a checkpoint. And until that checkpoint is met, then that cell will stay in a G1 phase. And in G1 phase, we've got a major portion of the cell's life. It's doing its normal functions and that sort of thing. If a cell cannot pass the G1 phase checkpoint, then technically we say that that cell is in a G0 phase. Like your muscle and nerve cells, they don't ever get out of G1, so we put them in G0. Although technically they're setting in G0 and G1 are no different. It's just the ones that we say are in G0 never make it out of G1. Near the end of the G1 phase, though, for those that do make it out, is the first checkpoint. This checkpoint has to be met by G1 cyclones and another protein that we say is CDC2. When G1 cyclones and CDC2 are both present in the cell, what we have found is that they actually fit together, and when they come together and lock together, they make this molecule G1-CDK. A G1-CDK is an enzyme that will increase activity for DNA replication to take place. So as G1-CDK complex is formed, then you will see that increase the enzyme activity for DNA replication, which is what's going to take place in the S phase. We've already found that out. So for it to leave G1 and go to S, the first thing that has to happen then is for the enzymes to be present to replicate the DNA. So then once the DNA is replicated, it goes through S phase and then it goes into G2. We've talked about G2 as just that second growth phase where it's getting bigger, getting ready to divide. There is a G2M checkpoint then that has to be fulfilled. The DNA has been replicated, the cell is grown, getting ready to divide. An increase now in MPF is triggered. MPF is a maturation promoting factor, which is what it was first called. Now a lot of times it's referred to mitosis promoting factor. But it triggers the release out of this checkpoint. So as the cell is going through G2, more growth factors are tripping those receptors, just like what we saw in the first slide. And in this case now, what they're triggering to be increased is the mitosis promoting factor. As this is increased, then this checkpoint will be fulfilled and it will trigger mitosis to begin. DNA replication is checked before this happens. So before MPF can really get at the concentration it needs to go into mitosis, DNA replication will be checked. And if something is wrong with the DNA, it will be halted right here until that problem is fixed. And we've actually done this in a lab where you have damaged the DNA so to see what's going to happen. And when that damaged DNA in that cell is trying to go from the G2 to the M phase, what we found out is that it won't go. It will stay there in G2 until it's able to fix the DNA, or at least give it a good shot at fixing it, thus to reduce mutation, stuff like that in future generations. So there is a replication check that takes place, and then you have the increase in the mitosis promoting factor, and that will send it past this G2 checkpoint and send it on into mitosis. Once it's in mitosis, there's one more checkpoint that it has to fulfill, and that is called the spindle checkpoint. In the spindle checkpoint, all the chromosomes line up at the equator of the cell in metaphase, and as those chromosomes are lined up on the equator, they should have spindle fibers connected at the kinetochores that we saw earlier in another lecture. So those spindle fibers are connected. As those are connected, the cell will release anaphase promoting factors, or APF, and this occurs when all chromosomes are attached to spindles. So as those chromosomes are attached to those spindles and they're lined up in the middle, that attachment gives off the growth factors needed to make anaphase promoting factors. Anaphase promoting factors will then trigger the beginning of anaphase. So anaphase won't start until you have APF, and APF is not produced until those spindle fibers are connected to all those chromosomes. Now the important part there is that, remember, in anaphase you're going to have the centromere divide and those chromosomes are pulled to opposite sides. So if those spindle fibers are not connected together to all of those chromosomes, then when the centromere divides, it won't be able to pull them that way. So you have to have that for that to take place. Alright, so the regulation of cell division is controlled. Here is a comparison of two of the levels of proteins that are involved in it. So when you look at this process, you can see on each stage so far that we're talking about how the control takes place. The amount of two important proteins can easily be graphed below. We've got the difference between MPF, the mitosis promoting factor, which we've already talked about, and cyclones. So as you can see, mitosis promoting factor, very low amount or concentrations of that protein through the G1S, G2. At the end of G2, remember, go back to the couple slides and you'll see where the mitosis promoting factor is. In G2, you start to get an increase of mitosis promoting factor. When that begins to increase, what we notice is that that's also when cyclones begin to become prevalent and increase. And there's a connection between these two because they will actually link together to do their function in mitosis, to promote the onslaught of mitosis and the things that are involved in mitosis and cytokinesis. And then you'll see the cyclones will drop off as the MPF also decreases. So as MPF increases, cyclones begin to increase because of their connection. And then as one decreases, the other obviously will decrease as well. And the presence of them is what dictates the beginning of mitosis and then the ending of cytokinesis. The last thing we're looking at is what happens when control is lost. If this process is a very controlled process, then what happens whenever that process isn't controlled? Well, you get cancer. Cancer is unrestricted and unrestrained, uncontrolled growth of cells in humans. So that mass of cells, that's a tumor, and that is cancer, and that's just a growth of cells that's not being controlled. A couple of genes that are associated with those, the P53 gene, P53 proteins monitor correct DNA replication. So it's one of those that's involved before the G2M checkpoint is met, is the presence of these P53 proteins controlling replication. So this will also halt the cells at the G1 checkpoint before they ever even get into the S phase. So if they're not present, then it will halt, or if they're in low amounts, it will halt there. Then once it goes into the S phase, these proteins will be what helps that replication take place before it can pass that next phase as well. If you have a mutated P53 protein, you have no halting abilities, and thus then uncontrolled cell growth. Proto Oncogenes are also another gene that's related to uncontrolled cell growth. These are mutated proteins that are always turned on to encourage cell division. They cannot be turned off. So they've been mutated to where they should be able to be turned on and turned off, but the mutation has allowed them to always be turned on, and that is then allowing that cell to always pass those checkpoints and thus produce these cells at a very rapid pace. Well, cells being produced at a rapid pace, you miss some of the other checkpoints of making sure the DNA is okay, making sure the cell's big enough to divide, making sure it's needed because of growth factors, stuff like that. Well, with these always turned on, it bypasses all those and just produces those cells. And that mass of cells then becomes cancer.